Because of breakage of the DNA strand during transfer, only a part of the donor chromosome is transferred. Since only part of the chromosome is transferred, it can not replicate in the recipient cell. Therefore, donor genes normally cannot be detected unless recombination between the incoming fragment and the recipient chromosome take place. Although high-frequency strains transmit chromosomal genes at high frequency, they usually do not convert F- cells to F+, or high frequency, because the entire F plasmid is only rarely transferred. On the other hand, F+ cells efficiently convert F- to F+, because the entire F plasmid is transferred. At some insertion sites, the F plasmid is integrated with the origin in one direction, whereas at other sites the origin is in the opposite direction. The direction in which the F plasmid is inserted determines which of the chromosomal genes will be transferred into the recipient first. The manner in which a variety of high-frequency strains can arise is illustrated. Use of various high strains has been possible of Escherichia coli to determine the arrangement and orientation of a large number of chromosomal genes, as will be described. Use of high-frequency strains and phenomenon of introverted mating. As is the case for any system of bacteria gene transfer, one selects recombinants from conjugation. However, unlike the situation is transformation and transduction, during conjugation both the donor and recipient cells are viable, so it is necessary to choose selection media where the desired recombinants can grow, but where neither of the parental strains can form colonies. Typically, recipient is used that is resistant to antibiotic but oxotroph for some substance, and donor that is sensitive to the antibiotic but is prototrophic for the same substances. For instance, in the experimental shown, high-frequency donor that is sensitive to streptomycin and contains wild-type genes encoding enzymes needed for synthesis of amino acids, threonine and leucine. And for utilization of the energy source of lactose, is mated with the recipient cell that is a mutant for these genes but is resistant to a streptomycin. For selection media, the minimal medium containing streptomycin so that only recombinant cell grow. The composition of each selected medium is varied depending on which genotype characteristics are desired, and the recombinant as shown. Understanding the mechanism of conjugation and knowing that the high-frequency strain is a cell with a F plasmid integrated into the chromosome should lead you to see that the transfer of chromosomal genes that will always be in the same order from a fixed site in a given high-frequency strain. The nature of the oriented fragmentary transfer of host genes during conjugation was first shown by a procedure called interrupted mating. The mating pairs are rather weakly joined and can be separated by agitation in a mixture or a blender. If mixtures of high frequency and F- cells are agitated at various times after mixing and the genetic recombinants scored, it is found that the longer the time between pairing and agitation, the greater number of genes of the high frequency that will appear in the F- recombinant. Genes present closer to the origin enter the F- first are also present in a higher percentage of the recombinants than genes that enter late, in addition to showing that gene transfer from donor to recipient is a sequential process. Experiments of this kind provide a method of determining the order of the genes on a bacteria DNA genetic mapping. The arrangement of gene loci on the chromosome is called genetic map. A transformation and transduction genetic recombination between high-frequency genes and F- genes involves a homologous recombination in the recipient cell. This has shown by the isolation of mutants F- strains that are unable to form recombinants when mating with high frequency. These mutants are Rec-A negative recombinant minus and are deficient in Rec-A protein because of the mutation in the Rec-A gene. It is important to remember that recombination is not the same as DNA transfer. DNA transfer is unaffected by the fact that the recipient is the Rec- cell, even though recombination does not take place after transfer. Transfer of chromosomal genes to the F plasmid. Occasionally integrated F plasmids may be excised from the chromosome, and the possibility exists from the incorporation at the time of chromosomal genes into the liberated F plasmid. This can happen because both integrated F plasmids and the chromosome contain a number of identical IS at which recombination can occur. Such F plasmid containing chromosomal genes are called F prime plasmids. These F plasmids differ from normal F plasmids in that they contain identifiable chromosome genes, and they transfer these genes at high frequency to replicate. F. Mediate transfer resembles specialized transduction in that only a restricted group of chromosomal genes can be transferred. Transferring a known F into a recipient allows one to establish diploids for a limited region of the chromosome. Such partial diploids are called merodiploids. This is important for doing complementation tests. Other conjugation systems. Although we have discussed conjugation almost exclusively as it occurs, Escherichia coli conjugation plasmids have been found in many other gram-negative bacteria. Indeed, conjugated plasmids of the incompatibility group. Incompatibility can be maintained in practically all gram-negative species, and DNA transfer between species in general can occur. Conjugated plasmids are also known in gram-positive bacteria. For example, streptomycin, enterococcus, and staphylococcus. As a rule, the mechanism of conjugation is very similar in the other gram-negative bacteria. To what we have discussed for the F plasmid, whereas conjugation involving gram-positive bacteria can be quite different. Some conjugation plasmids mobilize other genetic elements, as we have discussed for F plasmid, but this is not always the case. There are also elements called conjugative transposons that can transfer out themselves from the chromosome of the donor host to that of the recipient, and can also mobilize other genetic elements. Conjugative transposons have mostly been found in gram-positive bacteria. They have a variety of wide range and can be involved in the gene transfer between bacteria and different genera. The mechanism of conjugation used by these elements is not completely known, but it does seem to involve circular plasmids like intermediates. Several conjugated plasmids have also been found in sulfolibus. Very little is known about conjugation in sulfolibus, although it is known that cell pairing occurs before plasmid transfer and that transfer is unidirectional. However, with one exception, the genes involved seem to have little similarity to those in gram-negative bacteria. The exception is a gene similar to TRAG, whose protein product in F plasmid-mediated conjugation seems to be involved in stabilizing mating pairs. It thus seems likely that the mechanism of conjugation in archaea is quite different from that in bacteria. Complementation. The methods of bacteria gene transfer we have discussed in the preceding sections involve the transfer of only a portion of the donor chromosome. Therefore, unless recombination takes place with the recipient chromosome, the incoming donor DNA will be lost because it cannot replicate independently. In only two instances have we seen that a state of partial diploidy can be stably maintained. One was in the case of a specialized transducing phage, where the donor genes are maintained as part of the viral genome. The other wall was the use of F plasmids where the donor genes have become a part of the F plasmid genome. Since it is possible to create specialized transducing phage or recombinant plasmids using recombinant DNA techniques, it is now possible to put essentially any portion of the bacteria chromosome on the phage or plasmid. This can be quite useful for a number of purposes on being the complementation test. Complementation test. When two mutant strains of genetically cross-muted homologous recombination can yield a wild-type recombinant unless both of the mutations include changes in exactly the same base pairs. Therefore, if two different tryptophan Escherichia coli strains that require the amino acid tryptophan in the medium are crossed and tryptophan recombinants are also obtained, it is clear that the mutations in the two strains did not include the same base pairs. However, this experiment cannot detect whether the mutations were in the same gene. This can be determined by a type of experimental called a complementation test. Complementation was first used in a diploid eukaryotic organism. In diploid organism, the cell has pairs of chromosomes, one member from each parent. When mutations are present on the separate members of a pair, that is, one mutation from each parent, they are said to be a transconfiguration. On the other hand, if the two mutants are on the same chromosome, both from the same parent, then they are said to be cis-configuration. As we have discussed, true diploidy does not exist in prokaryotes, but a diploid state can be achieved for a region of the chromosome by transfer of chromosome fragment from a donor. However, the principle of the test and the non-collateral cis and trans is the same. If one of the tryptophan genes has been inactivated in a particular strain of the mutation leading to a tryptophan phenotype, then putting a copy of the wild-type gene from another strain into the same cell should restore the wild-type phenotype of the cell. That is, the resulting partially diploid cell. This will be true unless the mutation. One could then say that the wild-type gene complements the mutation, even though in prokaryotes only a chromosomal fragment is transferred. This fragment will typically be large enough to contain many genes. Remember that the genes for the biosynthesis of tryptophan from an operon, and therefore one can easily transfer the entire operon from one donor cell. Note that even if the operon in the donor also has a mutation, it will complement the mutated gene in the donor. If the two mutations are in different genes, the two mutations are then said to complement each other. This is shown grammatically. Mutations can complement because the genes encode proteins that can diffuse through the cytoplasm of the cell. One cannot complement mutations in regulatory sites such as promoters because these function at the DNA level. If each homologous DNA molecule